Psalm 95, verse 2. Let us come before him with thanksgiving, and extol him with music and song. Welcome to the lecture on neuroplasticity. In this first part, we're going to talk about some of the cellular processes that take place during neuroplasticity. In this part of our lecture, we will use the abbreviation LTP for long-term potentiation. We will also once again see the chemical symbol for the ion calcium, Ca2+. The CNS is constantly changing. It changes in response to the environment, and it changes in response to experience, as well as to illness and injury. Some of these changes involve a change in function of the neurons. This is known as functional reorganization. You can have changes in the amount or even the type of neurotransmitter produced and released. And you can have changes in structure, such as collateral sprouting and dendritic arborization. We will talk more about these specific changes throughout the next couple of lectures. I first want to talk about changes that take place regularly under normal healthy conditions. The first two of these are predominantly going to affect synaptic strength. The second two not only affect synaptic strength, but also affect the actual physical structure of the neurons. These changes include sensitization, short-term depression, long-term potentiation, and long-term depression. The term potentiation means an enhancement, so to enhance something. Cramming is a perfect example of how we sensitize the nervous system. We potentiate synaptic transmission or enhance synaptic transmission. We do this by having the presynaptic terminal increase the number of synaptic vesicles it contains so it can hold more neurotransmitter, and to release a greater number of neurotransmitter molecules when the action potential comes down and causes opening up of the calcium channels. More neurotransmitter in the synaptic cleft has more chances of binding to receptors, and therefore we end up with a greater synaptic response. So when we activate the presynaptic neuron, we get an enhanced or potentiated response in the postsynaptic cell. Importantly, this is reversible. So when you cram for a test, you might do very well on that test. But if I asked you those same questions 2 days later, you won't do so well because you've already started to reverse the sensitization and you've lost some of that information. So you don't really want to cram. You want to make sure that you are able to do a different form of potentiation that we'll talk about shortly. Another type of readily reversible synaptic change is called short-term depression, also known as habituation. Short-term depression can occur when there's lack of activation at a synapse. It can also occur with prolonged exposure to a benign stimulus. This would be something like a train on a railroad track. If you live near the tracks, after a while you don't even hear the train anymore. Or in my home, we have a number of older clocks that chime. Most of the time, I don't even hear the chimes. But when somebody who is not familiar with the clocks comes to the house, they hear them every time the clock goes off. Exposure to a very strong stimulus can also result in a short-term reduction in the sensitivity or response at that synapse. With short-term depression, we have attenuation of synaptic transmission. Attenuation means a reduction. This occurs both presynaptically and possibly postsynaptically, although postsynaptic changes don't always take place. Presynaptically, we either have a decrease in the release of neurotransmitter, or if it's a very strong stimulus or a repetitive benign stimulus, we can actually deplete neurotransmitter. You only have a certain amount of neurotransmitter and it takes time to synthesize it. And so if you're using it all up, you may be using it faster than you're producing it. We also see a reduction in the amount of intracellular calcium, which means that less neurotransmitter gets released from the axon terminal. If postsynaptic changes do take place, these are actually changes in the receptors themselves, so that the receptors are less sensitive to the binding of neurotransmitter. So while there'll be some impact and some opening up of ion channels, you won't see as much opening or for as long as you would have prior to the postsynaptic changes. In the presence of short-term depression or habituation, activation of the presynaptic neuron results in a decreased response in the postsynaptic cell. A more permanent change in synaptic effectiveness occurs with long-term potentiation. Long-term potentiation requires repeated calcium influx into the postsynaptic cell over time. Long-term potentiation has two basic phases. In the early phase, we move beyond the presynaptic changes associated with sensitization to add some postsynaptic changes. The repeated influx of calcium into the postsynaptic cell causes the postsynaptic cell to increase its production of receptors and inserts these active receptors into the postsynaptic membrane. An increase in the number of active receptors in the membrane is called upregulation. Because there are more receptors now and their associated channels to capture the neurotransmitter in the synaptic cleft, the result in the postsynaptic neuron is going to be enhanced. As repeated calcium influx continues, we move into the late phase of long-term potentiation. Here, the continued repetitive calcium influx activates genes in the nucleus of the postsynaptic cell. And these genes are important for the production of dendrites and dendritic spines on that postsynaptic cell. So the postsynaptic cell undergoes dendritic arborization, the development of additional dendrites and dendritic spines. So there are more places that synapses can take place. The increase in the dendrites, the dendritic arborization, has an effect on the presynaptic cell, stimulating it to produce more axon terminals. As a result, completely new synapses are formed, and we undergo what's called synaptogenesis, the birth of new synapses. Synaptogenesis and long-term potentiation occurs throughout our lifetime. It is strongest when we're young, but it never stops. So even older adults are capable of learning and are capable of adapting and changing. Synaptogenesis has been shown to be improved by physical exercise. So it's very important while you're in school that you take time to make sure you get your exercise in. It also requires, as I mentioned, repetition. We need to get that repeated influx of calcium over time. With this, it's best for you to do a small amount of something several times than to do a very large amount just once. The example I give here is you want to study for an hour for 10 days rather than study for 10 hours in the day before the test. Not only in the 10 hours, are you not getting the required repetition that you need? You're also not giving the neurons opportunity for resting. That leads to neurotransmitter depletion. And neurotransmitter depletion, instead of leading towards synaptogenesis, actually leads us towards habituation. So study all of your classes a little bit every day. Some classes more than others, but you need to study all your classes every day, not just study and cram the day or two before the tests. Long-term depression is going to be, again, involving structural changes, but this time in the other direction. Here you have established synapses, and you start to take away their parts and pieces. This can occur because we've had decreased firing of a synapse for a long period of time. You don't use it, you lose it. Or it could be you have a very strong stimulus that's happening over and over and over and over again. We basically monitor, if you will, the intensity of a stimulus based on the frequency of action potentials generated. Recall, we can't change the size of an action potential. An action potential either happens or doesn't, and when it occurs, it's always the same size in that particular neuron. So I can't have a higher intensity stimulus create a bigger action potential. Instead, what it does is it creates more frequent action potentials. If I have a high frequency of action potentials because I've got this really strong stimulus and I keep having it over and over and over and over, the neurons again will protect themselves from too much activity by starting to take away parts of the synapses. This can include removal of receptors from our postsynaptic membrane. It can also involve turning off of genes that are required for synaptogenesis, so we certainly can't make new synapses, but also for proteins that are important for maintaining synapses. So we start to actually take synapses away. Long-term depression and long-term potentiation are both more permanent types of changes, whereas sensitization and short-term depression are reversible, more temporary changes. Long-term depression can go to the extent of pruning, or pruning can occur because a synapse failed to form to begin with. Pruning involves reducing the number of synapses, taking away dendritic spines, removing a full dendrite, or even eliminating an entire neuron. Neurogenesis, the process by which stem cells become mature neurons, is currently only found functionally in two areas of the brain. One of these is the hippocampus, a structure in the medial aspect of the temporal lobe, which is very important in learning and memory. The other involves stem cells in the lining of the walls of the lateral ventricles. As these stem cells develop into immature neurons, the neurons migrate to become mature neurons in the olfactory bulb. There are stem cells in other areas of the brain that do develop into immature neurons. However, the environment in these other areas prevents the immature neurons from surviving to maturity and becoming mature neurons, making connections with other neurons. Currently, research is ongoing in how to provide the necessary environment to allow stem cells to survive all the way to maturity in the presence of injury or disease. All neurons require sufficient active synapses to survive. Neurons that fail to make a synapse or fail to have sufficient activity of synapses will be pruned away. The process of maturation of stem cells to mature neurons is affected by the internal and external environment of the individual. Some medications, such as chemotherapy drugs, can interfere with neurogenesis in the hippocampus. And patients who are undergoing chemotherapy will often report memory issues, something known as chemotherapy fog. Chronic intractable stress also retards neurogenesis, particularly in the hippocampal and perihippocampal area, the area surrounding the hippocampus. On the opposite end, neurogenesis is enhanced by regular physical exercise. It is also enhanced by an enriched environment. Individuals that are in very stark environments with little ability to interact with sensory stimuli or other individuals show a reduction of neurogenesis compared to individuals that have enriched environments with various types of sensory stimuli and interactions with others.